So now we're going to have a look at how mitochondrial defects can also cause disease in humans. And we're briefly going to talk about why that is. So endosymbiont theory that I'm sure you went over in high school. A brief explanation. Eukaryotic cells, so cells with a nucleus, have extracellular genomes contained in mitochondria and chloroplasts as a result of endosymbiont theory, where it's thought that a eukaryotic cell, like very long ago, partially phagotized the prokaryotic cells, mitochondria and chloroplasts, on two separate events. We have evidence of this from mitochondria and chloroplasts within cells having a double membrane, which we would expect if they were kind of engulfed from phagocytosis, which is shown on the bottom right, where the cell is basically reaching out and engulfing the other cell and kind of incorporating it within it. And DNA in these prokaryotes are contained within the nucleoid. So the mitochondrial nucleoid is a circular genome with 37 genes, lacking introns or gaps because the mitochondria is significantly smaller than the host eukaryotic cells, so they have to be a lot more efficient with their genomes. They have 13 genes for phosphorylation, 22 tRNAs and 2 rRNAs. Mitochondrial DNA is always maternally inherited because you get all your mitochondria from the egg, because the sperm lack mitochondria. So if you think about formation of the zygote, all your mitochondria are coming from the egg or the secondary oocyte and not from the sperm. This maternal inheritance of mitochondria has provided evidence that Homo sapiens and Neanderthals are not evolutionarily distinct. Despite being small, defects in the mitochondrial DNA, so in the nucleoid, can cause some severe human diseases, such as Lee syndrome and Leber's hereditary optic neuropathy, which we're going to talk about a bit now. So first, Lee syndrome, a disease that starts in the first year of life, so very young, characterized by a decline in mitochondrial function, marked by the symptoms seizures, altered states of consciousness, dementia, and ventilatory failure. So just keep in mind it's mainly affecting the central nervous system, and because it's affecting the central nervous system, you're having symptoms in cognitive abilities as a result of that. So as a result of it primarily affects the central nervous system as a result of mitochondrial defects in genes responsible for the electron transport chain, also called oxidative phosphorylation, resulting in insufficient amounts of ATP being produced. So I'm sure you remember from high school, the electron transport chain is responsible for producing the vast majority of ATP that we actually get from aerobic respiration. So if there's something wrong with the electron transport chain, you're not going to be getting enough ATP to be able to sustain yourself, and especially to sustain cells that have high metabolic demands, like nerve cells in the central nervous system. This deficit in ATP results in the symptoms listed above associated with progressive neurodegeneration. So, because you're not producing enough ATP, eventually nerve cells are going to die, and that's going to be a progressively degenerative thing that's going to happen throughout life. So as you get older, more are going to die, and then it's just going to get worse. Treatments for the syndrome are limited and depend on the specific genetic defect of the patient. So say there's a defect in a specific protein or coenzyme or something required for the electron transport chain. If you can supplement that through their diet, and the mitochondria can kind of incorporate it to do some successful oxidative phosphorylation to produce a lot of ATP, you can kind of combat some of the effects, although for a permanent cure you would have to edit their mitochondrial DNA. So moving on to Leber's hereditary optic neuropathy, as the name suggests, it is an eye disorder characterized by progressive loss of central vision due to degradation of optic nerves in the retina. This vision loss starts in early adulthood and is also caused by mitochondrial mutations similar to Lee syndrome. These dysfunctional proteins evolved in the electron transport chain that are going to be producing the ATP that the body needs to fuel these these cells that require a lot of ATP to actually function. So specifically, production of a dysfunctional complex 1 protein required for the electron transport chain results in much less of this ATP being produced, which causes optic degeneration in patients as the retinal ganglion cells have high metabolic demands that can't be met as a result of not enough ATP being produced because of this dysfunctional complex A protein. So these are two examples of defects in the mitochondrial genome, so the nucleoid, altering the general larger phenotype of the person because they're not producing enough ATP to support their cells that require high metabolic demands, so they're suffering because of it. But if we were to take these hereditary mitochondrial defects, which these patients could pass to their offspring, although this won't cure it in them, we can prevent their offspring suffering from the same mitochondrial disorder via use of in vitro fertilization to produce what we nickname a 3-parent baby, where the mother who suffers from the mitochondrial condition will have her nuclear material inserted into an enucleated egg from a donor containing healthy mitochondria that don't have the mutation in the nucleoid responsible for the mitochondrial disease. And the father's sperm will fertilize this egg containing the mother's DNA and the donor's mitochondria to produce a baby with DNA from the mother and father, as well as mitochondrial DNA from the donor. And I've put images in here of what the diseases actually look like because it's important to keep that in mind when you're actually researching this stuff, that they're real people with these diseases, and it's our job to kind of solve how to help